This is the fourth video of the second lecture for ionotropic receptors. In this video, we will focus on discussing NMDA receptors, their structure, composition, and function. So, as we've previously mentioned in other videos, NMDA receptors are one of the third glutamate ionotropic receptors. And because of their name, they're also considered the NMDA type of receptors. This is because these receptors receive their name due to their selective activation by the N-methyl D-aspartate molecule, or NMDA, which binds selectively to this receptor, activating its function. These receptors are very important for controlling synaptic plasticity and memory function. Activation of NMDA receptors results in the opening of an ion channel that is non-selective to cations. While the opening and closing of the ion channel is primarily gated by ligand binding, the current flow through the ion channel is voltage dependent. It is activated when glutamate and glycine, or also D-serine, bind to the ligand binding domain, depicted here. Different subunits of the NMDA receptors can either bind glycine or bind glutamate, but not both at the same time. And once this happens, they allow the ions, the positively charged ions, to flow through the cell membrane. However, for this particular type of receptor, extracellular cations such as magnesium and zinc ions bind to specific sites of the receptor, blocking the passage here of the different ions through the pore, and allowing a voltage-dependent flow of sodium and small amounts of calcium ions into the cell, and also potassium out of the cell when the magnesium and zinc are removed. Therefore, two events are needed for the NMDA receptors to open. One is the binding of both glutamate and glycine, and the second event is the membrane depolarization for the removal of the magnesium and zinc to finally allow the flow of cations. And we will see this in a little bit more detail in the next slides. Then this results in a calcium flux that this is very important for mediating synaptic plasticity and cellular mechanisms for learning and memory. As we just mentioned, glycine binding in the NMDA receptors is a key binding site to allow the NMDA receptors to work. So if NMDA receptors are only binding to glutamate, they're very poorly activated. So both glycine and glutamate must bind to the NMDA receptor for opening. And the concentrations of glycine are really small, in the nanomolar range, and very small amounts are required for binding to the channel. Then during normal conditions, or in most conditions, glycine binding site is occupied by glycine, so then you only need glutamate to bind. But in some conditions, glycine may also regulate NMDA receptor activity by binding or unbinding. And as we also commented in the previous slide, the serine, it's another amino acid that can bind to the NMDA receptor, which even higher affinity for the glycine binding site than glycine has. So this is another way of modulating NMDA receptors. And interestingly, glial cells release these serine. So it's being proposed that perhaps glial modulation of NMDA receptors are mediated through the release of these serine. In this diagram, you can see in better detail where the different molecules, ions, neurotransmitters bind within the receptor. as we can see here in the ligand binding site, there's a zinc molecule binding. Also from the inside, close to the segment number 2, there is a magnesium binding. We have also in the outside part of the receptor, there is the glycine and other substances binding sites. This receptor is also susceptible to different modifications that modulate activation and desensitization as well, such as glycosylation and phosphorylations. And here are some examples. Usually phosphorylations occur in the intracellular space, while glycosylations are occurring in the extracellular space. And then here you can see how different agonist and antagonist sites can be found. Then regarding the opening and closing of the NMDA receptors, and as we've already talked about, this is a very complex process. If we compare with the AMPA receptors in the top part of this diagram, AMPA receptors only need the neurotransmitter to bind to cause the opening of the channel and the ions to flow through the channel. In the case of the NMDA receptors, it's briefly depicted in the bottom part. Then when the receptors are closed, they do not allow the flow of ions. However, upon binding of the neurotransmitter, glutamate and glycine, then the channel opens, but it still does not allow ions to flow through the channel due to the blockage by magnesium and zinc. And then it requires not only binding of the neurotransmitters, but also a depolarization of the membrane to fully unlock the channel, remove these magnesium and zinc ions, and fully allow the cations to go through the channel. Here there's a little bit more detail about how this removal of the magnesium happens in a voltage-dependent manner. When the membrane potential is about minus 60 millivolts or more negative, even if there is neurotransmitter binding, the channel keeps blocked. It maintains in a block state through the magnesium. And the membrane potential needs to reach at least minus 30 millivolts or more positive potential to allow the magnesium to leave the receptor to fully open the channel. And this is just a summary of what we've been talking about. So the NMDA receptors conduct only when two things are present. One is that the glutamate is present and that the membrane potential is more positive than minus 30 millivolts. So then you will need an action potential plus glutamate plus the removal of magnesium to cause a fully opening of the channel. Regarding NMDA receptors pharmacology and with a special emphasis in antagonists, there are several molecules that either work as a competitive agonist, as a competitive antagonist, or they just indirectly block the receptor by blocking the opening of the channel. So in the first group, the competitive antagonist, we have the AP5 and AP7. They stand for 2-amino 5 or 7 phosphonoheptanoic acid. And these substances are used to antagonize NMDA receptors. Some of them, such as the AP7, have anticonvulsant properties, but they can also, depending on where they're injected, they can mediate muscle rigidity or catalepsy. And there are other competitive antagonists of the NMDA receptors, such as ketamine, that have hallucinogenic effects. On the other group of antagonists, those that indirectly block the channel or inhibit the channel by blocking the opening, include phencyclidine and disocelpine. And those two molecules act as if they were cooperating with the magnesium chloride to block the opening of the channel. And their use can also have hallucinogenic properties. Then, as we've seen earlier for the AMPA receptors, the NMDA receptors are also susceptible to alternative splicing, which, as we've mentioned earlier, provides more versatility to the system. Besides the different subunits that we can, or the different isoforms of the subunits that we can find within the NMDA receptors, there are also alternative splices for all of them. And they can be generated up to 2 different splicing isoforms for every subunit. So that generates a total of 8 different variants in subunit composition. Then NMDA receptors have also need to be inactivated or desensitized after they've been activated. And there are 3 known mechanisms that are responsible for the desensitization of the receptors. There is internalization of the receptors via endocytosis mechanisms of the receptors into the intracellular space. Well, they can undergo different processes such as recycling back into the membrane or degradation into late endosomes. So then the proteins of the receptor are degraded. There are also other mechanisms such as phosphorylation. We saw in the previous slide that the intracellular part of the receptor is susceptible to different phosphorylation sites, and those can also mediate the closing of the receptor and the inactivation. And then there are other processes that are not depicted in this diagram but are also responsible for desensitization of the NMDA receptors, such as activation of second messenger systems. And we're not going to discuss that, but those are also very complex, and it may include other G protein coupled receptors proteins to mediate the desensitization of the NMDA receptors. Overall, all these mechanisms lead to a slow and long-term reduction in signal amplitude of the NMDA receptors, and this is also very important to mediate synaptic plasticity. And finally, as we also mentioned for the AMPA receptors, localization of glutamate receptors need to be very finely controlled, so they are located at the sites of synapses. There is certain lateral diffusion. They're mostly anchored to the membrane at the PSD by different scaffolding proteins. In the case of NMDA receptors, they directly bind to a postsynaptic scaffolding protein called PSD95. And this PSD95 protein has a lot of different domains that are called PDZ, which are responsible for interacting directly with the subunits of the NMDA receptors. And then this PSD95 binds to other proteins that end up binding to the cytoskeleton, and that's how the entire complex is anchored to the membrane. Other ionotropic receptors such as AMPA and kinase receptors, they can bind PSD95, but it's been supposed that there is a more indirect mechanism via another protein called estargacin. And this estargacin is a transmembrane AMPA receptor regulatory protein that it will do the same thing as I just described for NMDA. It interacts with the PSD95 protein, which helps to interact with a cascade of different proteins that link the receptors to the cytoskeleton. And finally, this is just a very brief list of The biggest differences between AMPA receptors and NMDA receptors. If you remember from our previous video, AMPA receptors are permeable to sodium and potassium and not much to calcium except for those subunits that are specifically spliced and edited, such as the GluR number 2. Then these receptors have also a very low affinity but with a very fast activation. There is also a fast deactivation or desensitization due to the intrinsic properties of the receptor and how they can create a conformational change that is intrinsic to the receptor and mediates its own desensitization. And these receptors are mostly associated to a fast synaptic transmission. On the other side, what we just saw in this video is that the NMDA receptors are permeable to all three cations, sodium, potassium, and calcium. On the other side, they also have a very high affinity for their neurotransmitters, but the activation is slower since there are different mechanisms that have to be coincident to fully activate these NMDA receptors. They're also susceptible to a slower deactivation because it requires additional mechanisms that are not intrinsic to the NMDA receptors, such as phosphorylation, internalization, or second messenger activation. But this situation is key and crucial for mediating synaptic plasticity. And as usual, I leave you here with a short quiz where you can revise the most important concepts that we've seen in this video, and we will discuss these and other questions in class.